What's the most interesting thing that's been happening in the last couple of years that's really made pulmonology a hot topic?
Lung cancer is now a disease that is not, let's say, a group of patients who have lung cancer. No, it's now all types of diseases. Yeah. It's going to be more and more separated in subgroups based on histology, based on molecular markers, based on targets for drugs, whatever. So that's the future. Yeah. That's going to change our treatment and the outcomes as well, I hope. Yeah. That's one thing. And the other thing is, what we as medical doctors have done much better is integrating our efforts. So coming to combined modalities, combining doctors who give drugs, who give radiotherapy, who do the surgery, who do the endoscopy, things like that. I think that is much, much better than it was 30 years ago when we met.
Sure. Yeah. And your group in Vrije University in Amsterdam, what are you working on?
We work on, my group is especially working on two areas. One is pulmonary circulation, but is not related to lung cancer, but a lot of mechanisms are in common. Sure. Angiogenesis, yeah. Pulmonary circulation has a lot of angiogenesis as well, as has lung cancer, so that is the common area. We mainly work on treating a lot of patients in combined modality trials, new drugs, using trials for that, and another part is looking at very early stages of lung cancer. For instance, the pre-neoplastic stages detected in patients who come and we find during bronchoscopy squamous metaplasia and follow up these patients and see whether they develop lung cancer or not and look at the tissue that is obtained then. And we have tissue from up to 15 years ago and follow up with these patients. And we can, hopefully we can find out the markers that are important for the ones who are going to develop lung cancer and the ones who are going to not.
And this is in heavy smokers?
This is in, yeah, the majority of them are heavy smokers because early, let's say, detection of early lung cancer is always in the central airways. Sure. It's not in the periphery of the lung. It's impossible to get the tissue from that. That's another area we're working on. We're trying to set up a system in which we combine CT, PET, and endoscopy, virtual bronchoscopy, and guide us to the lesion in the parenchyma. But that is still the future. I think that's an area that might be very interesting to get tissue cells from areas which you normally only get if you do surgery, but then your tumor is gone or your pre-neoplastic stage is gone because you have taken it out. So you never know what is the real thing that's going to develop. How does a type A Noguchi develop to type G? I don't know.
Sure. Yeah. Chemo prevention studies, are they on the horizon in this group of people who are at risk and whom you've proven dysplasias and so on?
I think if you know the target, then the time has come to do the chemo prevention. So far, I don't know. I really don't know because we need to know what are the abnormalities, in fact, in the early stages. For squamous, we come closer to that. For adeno, we don't know at all.
And targeting in therapy, EGFR and so on, that's sort of opened up a Pandora's box in lung, which is fast resembling breast cancer in terms of the subdivision.
Yeah. We're 15 years behind that.
Yeah. Where are you now? Which are the hot genes or proteins? Or
of course, what the TKIs can do to patients with the relevant mutation. It's a tyrosine kinase inhibitors. Sure. What they can do in the patients with the relevant mutations in EGFR, that is a thing that is a breakthrough, I would say, for this group of patients. But that's only a minority. Sure. To find these patients, these are very often non-smokers or long time ago stopped smokers. So different population than we used to see 30 years ago. But these patients, if you can find them, you have a much better option for treatment than we used to have. Now, what we are working on now is making specific PET tracers to detect these. So we have labeled erlotinib, can detect it with a PET camera, and can find in that way patients who have the mutation in EGFR.
Which label do you put on it? Gallium?
No, it's carbon, carbon 11.
Okay. So you make carbon-11 labeled a lot
Yeah,
in the lab,
yeah,
and then
yeah.
as we did with temozolomide a long time ago.
Yeah,
Sure.
yeah. And that's a thing that's going to have a much wider application for other drugs as well. As soon as you know the target, you know that you can make the drug that goes there,
Sure.
or whatever that binds to the target.
And here in Geneva, while this meeting's on, we're witnessing the launch of a PET MRI joined-up camera. Is that taking us one step further in the imaging?
I'm not sure whether that is for lung the most important thing, because MRI for lung is not that good.
Mediastinum, huh?
Yeah, maybe for the brain.
Yeah.
And a brain is a very common site for lung cancer as well, so that is a problematic area. I've been working on that for decades already. So
I recall you doing phase 2 trials a very long time ago.
Yeah.
Teniposide comes to mind immediately.
Yeah, etoposide, teniposide.
You are chairman of the IALSC meeting in Amsterdam next year. What are the highlights of that meeting going to be? We should all come to that, I think. IALSC
you should come there. If you're interested in lung cancer, from the beginning till palliative care, you should be there.
Good. Dates are July?
3rd till 7th.
Of July in Amsterdam.
Yeah.
Okay. So what are the big highlights that you're going to?
I think the main focus is going to be on the personalized medicine.
Right.
But that is, let's say, the commercial theme that is everywhere nowadays. So, but that's not going to change there. Yeah. It's going to be there as well. Okay. And we set up all kind of things for all people who are really interested. So not only medical oncology, pulmonary, but also imaging people, pathology people, molecular biology. We set up hands-on training sessions for young investigators to learn something from us in small groups. So it's going to be a nice program.
Thank you very much indeed. We'll hope to interview you in Amsterdam in July on eCancer TV.
You're more than welcome.